Hi everybody. My name is Pau, and today I'm going to talk about the project ABAMP. So whenever we are interested in characterizing environmental samples from a metagenomics point of view, we are basically interested in retrieving, analyzing the sample from the genomic composition of the samples. So as you might imagine, this step of actually retrieving the genome from the environmental sample is crucial since it's going to affect the downstream analysis. However, this step of retrieving the genomes is not is very challenging. Usually when we have an environmental sample, as you know, and want to sequence it, we have to break down the genomes present on the sample into the reads and then try to do all the way to the assembly and up to, if we can, to the genomes. Unfortunately, due to the biological complexity that we have in environmental samples, when we do assembly graphs from metagenomic samples, we end up with something like that, where due to sharing sequences between organisms, sequence repeats, mobile elements, each of those colorful stripes represents a contig, and each of those edges represents a potential connection of that contig with another contig. And that's why we need binning, which basically binning it's about try to group reads or contigs by its genome origin using different methods of techniques. In our approach, we decided to do metagenomics binning based on contig features. There are two main established contig features. One is the coabundance along samples. We are looking on the profile of abundances of contigs along the samples. So it's a correlation-based feature. And then we have the tetranucleotide frequencies, where that it has been shown that these kind of K-mer with length 4 tetranucleotide frequencies are shared along the taxonomic tree, so we can use this kind of feature for binning. And therefore it's a composition-based measure. Those features, they are is it good? That should be a green check. They are positive because they are not reference biased. That means that we are not using any kind of information from the references for the binning. They're complementary. They are somehow saying the same, but in alternative ways. As I said, one is correlation-based and the other is composition-based. They allow the multi-sample approach, right? Because if we use this kind of profile of multi-sample along the samples, as many samples as we have, as more clear is going to be this kind of profile of abundances. Unfortunately, they are noisy, and there is no straightforward way to integrate them. So to summarize the problem, we start from a huge set of contigs that is generated from a relatively small set of genomes. We have complementary noisy signals that connect those contigs from the same genome, in this case TNFs and coabundances. So it seems that it's the problem that deep learning could help us with. And it can help us with basically integrating those signals and denoise this signal. And we believe that a right approach could be to use variational autoencoders. The process we use here is to take these contig features, the coabundances along samples and the tetranucleotide frequencies, and use an encoder to actually project and learn these features and projects them into a latent space. I want you to think of this latent space as a latent space that's continuous, Gaussially distributed, meaningful, and clusterable. Now we have a decoder that actually samples from this latent space and aims to reconstruct the input. The idea is that since the decoders reconstructs the input only from the latent should contain the essential information from the input data. And that's how it works for metagenomics binning. We have our variational autoencoder, we have our latent space, and here in the downs, two plots, we have the two principal component analysis of the input data without any kind of processing, and the latent space, where each point is a contig and each genome, it's a color. And we can see how the latent space is quite more meaningful for binning. Therefore, the only thing we have to do is to apply a clustering algorithm developed in the group and to retrieve the genomes. And it seems to work. That was developed in the group and published around 1 year ago and called the BAMP, Variational Autoencoders for Metagenomics Binning. However, variational autoencoders are essentially generative models. But what that means is that once they are being trained, we use the latent space, or we use better the prior, and we use the decoder that has been trained to generate new data. Therefore, since the latent space looks kind of a Gaussian, it's not really optimal for clustering. It's not super easy to cluster. So since then, new models have been proposed, which could be more promising for our purpose, which is binning. And among them, one that we wanted to give it a try is adversarial autoencoders. Adversarial autoencoders, they are quite similar to variational autoencoders. We have our input data, our encoder, decoder, and reconstruct. But now we have two latent spaces. One is the Z latent space, which is holds kind of similar properties than before. It's a Gaussian distributed, continuous, clusterable. And then we have the Y latent space, which it's a categorical one-hot distributed latent space. So now in each forward pass, we'll have the encoder projects our contig in a continuous Gaussian distributed latent space into one hot vector as well. Another important difference between like adversarial and VAEs is that now to regularize these latent spaces, we have two additional networks, the discriminators, that are continuously competing against the encoder and the decoder for the regularization of those latent spaces. And that's why we call them adversarial autoencoders. And that's how we apply them for binning. As we said before, the Gaussian distributed latent space is just clustered, and the genomes are retrieved, like the AAEZ genomes. And since like the categorical latent space is like a space with one hot vectors, it could be just interpreted as a classification on the method of those contexts. Therefore, the only thing that we have to do is to retrieve the context from it and like to get the bins. And without like a lot of creativity, we call that EMP, adversarial autoencoders from metagenomics binning. Interestingly, when we were analyzing the composition of the bins reconstructed from those two latent spaces, we realized that there is somehow a level of complementarity. Here we have the latent, like the bins reconstructed from the Z latent space and the Y latent space. And see that there is some kind of like overlap as well, but some complementarity. Like it seems that each latent is capturing something different from the data. As well, if we add BAMP latent bins reconstructed from the same sample, we can see how also there is a level of complementarity. And that's why we decided to develop the pipeline that actually takes these context features, which are the same for both models, and runs in parallel BAMP and EMP, generates these three sets of bins, and dereplicates them into a unified dereplicated disjoint set of bins. And we call it ABAM, Adversarial Variational Autoencoder from Metagenomics. And it seems to work pretty good. This is like the results with benchmark data sets, where on the y-axis, we present the number of high-quality reconstructed genomes. Green represents MetaBat2, which is kind of like one of the popular like tools for binning. Blue BEMP, orange EMP, which is the only integration of the EMP latent spaces, and light pink represents ABAMP. And we can see how like we reconstruct on average 30% more bins than BEMP and 110% more bins than MetaBat on the benchmark data set, which are around like 200,000 contexts, 10 samples. In the same way, when we like check like the performance on real data sets, we kind of like see the same tendency, where like we obtain like slightly like more bins like than BAM and quite a lot more than MetaBat. Another thing is that like this increase of like a number of near complete bins doesn't come like with a super expensive resource time. As we can see here, we have like that, like which is like the running time of the different tools, BAMP, EMP, eBAMP, and MetaBat over the benchmark and the real data sets, and we can see that there is actually like an increase of the runtime, but it's not dramatically. Also, one like interesting aspect is when we were analyzing the taxonomic diversity or resolution of the bins reconstructed from one of those real data sets, the Almeida data set, we can see how ABAMP like expands like the resolution like of the species, genus, and family reconstructed from this data. One of the important points as well is that like whenever we are running like BAMP or ABAMP along multiple samples, we will have like some organisms that they have like different prevalence along those samples. And as we can see here, BAMP and EMP seem to like have like some kind of tendency to capture and reconstruct organisms they have like a different prevalence along the samples. And that's like kind of like a strong point to use these two methods altogether, because both of them, they seem to have like a tendency to reconstruct different parts of the data, which is like at the end, by dereplicating them, we get the benefit of all of it. Finally, we were also interested to see like what's the difference on the quality of the bins that they are reconstructed by both methods. And here, like we see like the taxonomic tree of life of one of those real data sets, the Almeida data set. On the blue stripe represents the bins reconstructed by both methods, but BEMP represents a higher score, and the red stripe the same, but where EMP presents a higher score. And we can see how EMP reconstructs or like provides a higher score of around 2,000 more near complete bins, which means that there is a significant increase on the quality of the bins reconstructed by EMP. Finally, as a take-home message, I just wanted to like to clarify that ABAM seems to like to reconstruct around benchmark and real data sets 30% more near complete than BAMP, which is kind of like the highest performance reference-free binner that it's out there so far, to our knowledge. There is a higher taxonomic resolution. We have an increase on the quality of bins. It's a pipeline that is parallelizable. It works for us for both long and short read data. And apart, we also like developed in the group classifier of the bins that allow us to classify which bins belong to genomes and which bins belongs to pages. And finally, I would like to just acknowledge my group, especially Simon and Jacob, and I will be happy to take some questions.
Thank you. Any questions from the audience? I'll start with one, since, yeah, great work, and congratulations to beat our tool. Yeah, MetaBat. So I have a question about the running time. So when you do the comparison, does that include the training?
Yes.
So from the training and prediction is still very fast with thousands of samples, right?
Sorry?
So when you train the autoencoder, so that the runtime include the training of the
Yes.
autoencoders. Okay, wow, that's very impressive. So the second question is, have you looked into those sample set with fewer samples, fewer number of samples?
The benchmark data sets are around 10 samples, and the real data sets are around 1,000 samples.
Yeah, but how about this a single sample data set?
It seems to work as well. I mean, of course, like the weight of the profile of abundances, it cannot take advantage of it as much as it can with more samples, but it performs.
Okay, thank you.
I have a question, maybe.
I think, yeah, there's another question, please.
Okay, so you showed data that your tool can produce more beans, which is great. Have you looked at the check-M metrics like the M50, not M50, sorry, the completeness and contamination, those metrics?
What I mean, like the thing is that like with benchmark data sets, it's pretty straightforward to evaluate it because we already know the truth, right? But with the real data sets, the Almeida and the Human Microbiome Project, that's how we evaluate. We only show near complete beans by check-M, that like it's about like a 0.9 recall, 0.95 precision.
Okay,
So,
thank you.